Hello, in this video we will look at the immuno-inflorescence direct and indirect and what is the significance of the immuno-inflorescence. So let's begin to understand the immuno-inflorescence without any wasting time. So in this method we use the antibody for detection of shape, size and structure of antigen. Antigen can be molecule, antigen can be bacteria whole, or antigen can be the segment of the cell or organelle. So it is visualization technique in which specific parts are stained and check the presence of antigen or part of molecules of the inside of the cell or outside of the cell. So we are studying the part of the cell segment which that is detected by the shape and structure and their size with immunoinflorescence antibody. So stain molecule for study the sample basically can be bacteria, DNA or microorganism or virus. You can say everything. So let's begin to understand. Here I am taking the cell of the human, which that is we are detecting the mitochondria of the human. If it is non-function or something that is disease causing syndrome due to the mitochondria, lack of the mitochondria in the cell, or their shape is disrupted. So here is a person which that is contain a tissue, and this tissue contain a nucleus and organelle. So this tissue is separate and put into the test tube as a sample. So the cell lysis by the mechanism of different mechanism to cell lysis or breakdown of the cell into the chunk small pieces. So these small pieces contain different densities. So we will separate the density by centrifugation or centrifuge that test tube. After the centrifugal force, that's on the basis of different density in the test tube, that is layer or phase will be created, and that's layer on the basis of the density of the molecule. It means the more dense molecule will move downward while the less dense molecule will move upward. So it means the different layers will be created. So that's we will check the whole test tube at different phase to get the sample, and this sample put into the slide or slide cover and put into the slide in the drop wise, and we will check the whole phases, and we will first find the mitochondria and where is the mitochondria. So remember about that. That is the biotechnological technique to understand the separation of the organelle or something. So we are checking the mitochondrial identification and mitochondrial study. So let's begin to understand. We will check the all phases, and we will found the mitochondria as organelle, while the other sample we will take a different sample by a different layers of the test tube on the basis of their density, and ultimately at last we will find the mitochondria. So the mitochondria is basically is not a complete shape. We cannot understand the complete shape. So here is the drop we will put into the slide and cover with the slide cover. So identification in the lab by a microscope. So after this light microscope, we can see the organelles, which that is the smaller pieces. So this organelle is a part of cell, and we will take all density sample for detection and select our desire. So we are selecting the mitochondria. So after this mitochondria, this is we will put in as a sample by the confirm the mitochondria, and this sample, the half sample or some segment of the sample will inject into the animal, and after this injection of the animal it is called antigen, and animal will produce the antibodies against this antigen mitochondria. So after this injection into the animal, that mitochondria will create a disturbance in the animal body. So the animal body will produce the antibodies against the mitochondria, and after the production of antibodies, it means the antibodies are present in the animal blood. So remember about that. So when the antibodies are present into the blood, so we will take the sample of the animal. That blood sample contain a many other type, also contain many other antibodies, but that antibody is not against the mitochondrial antigen. So it means we are getting the monoclonal antibody. So the monoclonal antibody is the another way. Monoclonal antibody is production by hybridoma cell technology also. So we are not understanding about the monoclonal antibody production by hybridoma cell technology, but you can watch another video. I will give the link in description below. So the blood serum sample will contain a different type of antibodies, but the mitochondrial antibodies is also present. So that antibody is our desire for checking. So these antibodies, basically whole antibodies, which that is different antibodies, we will linked antibodies, anti-immuno anti-mitochondrial antibodies, which that is we will fluorochrome stain, we will stain the fluorochrome or fluorescent antibodies. So these fluorescent antibody we will put the color producing fluorescent antibody. So these antibodies called fluorescent antibody after the staining and fluorochrome. So that staining mechanism is long, but here we will just understand about the mechanism. And here the no we have in fluorescent linked antibody or stain linked antibody, as well as mitochondrial as a antigen. So remember about that. We have two things. Number one is the inflorescent linked antibody or stain linked antibodies, and on the other hand, the mitochondrial as an antigen. So the two test tube are present in our lab. So let's begin to understand what is the basic mechanism about that. So important thing is that, so here we will put the antigen as a mitochondria, put into the media, culture media or something which that we are using. So the antigen mitochondria are present here. So the antigen antibody antigen complex is complex coating mitochondrial buffer, basically used for the buffering of the retaining of that antibody antigen complex. So antibodies we will add in fluorescent linked monoclonal antibodies or primary antibodies. So for example, there are a lot of antibodies are present in the sample. So that is mitochondrial, basically in fluorescent antibodies, which that is against the mitochondria will as an antigen will bind with the mitochondria to become a complex formation, antibody antigen complex formation. So after this, the other antibodies also linked with the color, but that antibody is not useful because we are getting more antibodies, but the specific antibody will bind with the mitochondria. On the other hand, the other other antibodies will be washed away after the washing process, and buffer is used for the retaining of that antibodies which that is desirable for us. So the buffer use and washing process to remove that wasteful antibodies or non-desirable antibodies. On the other hand, the antibodies which that is against the mitochondria will bind with the mitochondria as a primary antibodies, which that is attached with the immunofluorescent color, so that after the staining we will doing this process. So desired part of the cell, it produces light, is known as the inflorescence. So that after attachment of this antibody with the antigen is produce the color, and that's color we can detect that color and their shape, because whole antibodies around the cell or something are mitochondria, so we can check the shape and their structure. So the different fluorochromes produce a different color. When attached with antibodies, we can visualize more than one part of the cell. For example, here is the bacteria which contain a red color showing in the flagella, while the blue color showing the DNA and red color showing the pili or something. So the different colors show due to the different antibodies against this antigen when we will introduce the animal differently and separately, so we can produce many more antibodies. So I hope you make sense about the direct immunoinflorescence. This was the direct immunoinflorescence. I am recapping direct immunoinflorescence and indirect immunoinflorescence. So here is the directly bind with the antigen. So it means here is the A antigen, and that's primary antibodies will bind with the fluorescent, and that is known as a primary antibody which that is directly attached. But the major problem is that it is less prominent or less sensing mechanism, or less sensitive. So it means we are using the more sensitization or more light emitting, more sensitive reaction. So we can use that primary antibody again into the animal. So after that antibody we will take into the injection and move into the inject into the animal, and after this antibody play as an important role just like a antigen. So these antibody contain epitopes. So there are several epitopes against these antibodies. So that's several epitopes will bind will we, several epitope is play a role as an antigen. So this antigen is known this antibody is called antigen for that animal, and animal will produce antibodies again against these antibodies. So epitope bind with that antibodies which that is recently produced from the animal. After this we will take the serum from blood, and that antigen binds antibody with epitope of foreign antigen. So we are getting as a sample. So after this sample we have many more antibodies against the primary antibodies, which that is after the recognition of epitope are present into the primary antibodies to produce the against that epitope is known as the secondary antibodies. So these secondary antibodies will be removed by the taking of injection, and that's take out this sample, and inflorescent linked antibody bind with the secondary antibodies, and after this linking of the secondary antibodies we can detect that is the secondary antibody. But the non-inflorescent linked antibody is known as the primary antibody. So in this method we are not staining basically the primary antibodies, but in the direct method we are using the staining, but in this mechanism we are not using that strategy. What important thing is that we are linking the fluorescent with that secondary antibodies. So after the linking of these secondary antibodies, it means the one antibodies contain a more than one antibodies more to create a more light. So the more light emitting, more sensitive reaction. It means we can detect easily as compared to the direct immunoinfluorescence method. So the non-influorescent linked antibodies here, that is the primary. So important thing is that a last thing is that the uses in microbiology we will discuss in the using of the microbiology. So the important here is I am drawing the box to check that use in microbiology. So we can use DNA structure checking, DNA structure analysis, molecule structure, cell size, nucleus and chromatin or chromosome and organelles or something more. So you can see that are different molecules. But one more thing is important, that we will take the sample, and here we will go back, and here is the after the cell lysis. So here we will not cell lysis, we will take the sample and we will put into the paraffin wax embedded. So we will embed the paraffin wax and embed it to become a microtome. So that is the thin slice will be create a many more. So this thin slice is known as the microtome. So the transfer into the clean glass slice. So that's clean glass slice we will transfer and dewaxing by dewaxing agent to create that desirable molecule by checking due to the for example, after this slide we can check from the microscope to identify that molecule and put the again antibodies again. That process is again. So I hope you make sense about this video, immunoinfluorescence direct and indirect. So thanks for watching. Please make sure to subscribe, like and share this video in your friends.